We've been talking about phylogeny and taxonomy and how we use the evolutionary history of an organism along with descriptions of that organism, classifications of that organism, to help us to get a better understanding of the evolutionary relationships and biodiversity relationships of organisms on Earth. We're going to wrap up our discussion on taxonomy, phylogeny, and systematics by looking at a couple of mechanisms that cause genetic evolution, cause genetic change in a population. And we may have talked about this a little bit in chapter 19 when we talked about the mechanisms of microevolution. That's not quite what we're talking about here. We're talking about how large-scale changes and genetic transfers aid in the process of evolution. One way that we can follow evolutionary change is by looking at molecular clocks. So a molecular clock is basically using evolutionary changes at the sequence level to set a time frame for evolutionary events and evolutionary relationships. So researchers want to know how our different groups are related, and they also want to know when or how they branched off and how long it has been since those branch types. And we can infer that timing by looking at genetic sequences. As DNA replication happens, some errors are going to come up and there are going to be changes in DNA sequences. So mutations are going to arise based on our natural molecular processes. DNA polymerase works to copy DNA. In some organisms, there are mechanisms to limit mutations, but sometimes those mutations happen anyway. Some mutations are going to be favorable. These are ones that are going to accumulate in a population. Some of them are going to be detrimental, and those are going to be quickly eliminated from the population. Many of them are going to be detrimental if they occur in a coding sequence, meaning if they occur in a gene sequence that is doing something, producing a protein or a regulatory sequence or something in a genome that is going to affect the phenotype of an individual. But not all of the genome is coding sequence. Some of the genome is essentially a neutral region, meaning there is nothing being coded there. The DNA has no purpose, or at least it doesn't have a purpose as far as phenotype is concerned. So the neutral theory of evolution says that much of evolution of genetic sequences takes place as neutral changes in that genetic material. And the neutral theory of evolution says that along with those accumulations of neutral mutations, those mutations also occur at a relatively constant rate or an average rate across many different organisms. So if neutral mutations are occurring at a constant rate, then they can serve as a clock, a molecular clock, by which we can measure evolutionary time. There are some limitations to this. One is that not all organism sequences change at the same rate. So there's no single constant rate of of neutral mutation, but we can generally use this. But the more important part of this is that if we have neutral mutations accumulating, the more mutations that accumulate are going to represent a longer time frame since two groups split from one another. And conversely, less differences in those neutral sequences are going to suggest that those organisms diverged at a later date, more close to current time, and are therefore more closely related to one another. Again, this all goes back to accumulating changes and who shares those accumulated changes. So whether we're using physical characteristics, direct sequences, or neutral mutations, the more they share, the more closely related two organisms are. So this diagram represents that. So this diagram on the left shows the evolutionary time since divergence, so since the split from a common ancestor, and then the number of base pairs difference. And so generally we see as time increases, there's going to be an increase in the number of differences between sequences. On the right, we have time in millions of years. Down here, we have the earliest branching ancestor. Up here on the right, we have the latest branching ancestor. And then we have these color-coded bars. These color-coded bars tell us a relative difference in time since divergence. So we've got the Siamang here compared to the rest of our primates. And if we look at the accumulation of those random neutral changes, we see a whole bunch of accumulation compared to everyone else, suggesting that the Siamangs branched off much earlier than the others. There's no bars here for orangutans or gorillas, but we can then assume that we would have a shorter bar, a shorter time frame, a shorter amount of time since accumulation between the orangutans and the other groups. Likewise, less accumulation between the gorilla because we believe that they have separated much later. When we look at humans compared to chimpanzees, we have less accumulated differences, which suggests a split much more recently. And then between two different chimpanzee species, the common chimpanzee and the pygmy chimpanzees, or the bonobos, they have very few differences, relatively speaking, indicating that they diverged much more recently. And that makes sense. If you think about a common chimpanzee and a pygmy chimpanzee, they are very, very similar to one another. So it makes sense that what they would have diverged much more recently. Another mechanism of widespread genetic change is vertical evolution versus horizontal gene transfer. So vertical evolution is sort of our traditional view of evolution. This is where we get changes due to descent from a common ancestor. So in vertical evolution, we have changes that are passed down from parent to offspring over many, many generations. And that is where we're going to see most of the accumulation of genetic change, is by passing from parent to offspring, from a common ancestor to a derived organism or a new branch from that tree. But many, many genes are thought to have been acquired in populations by what we call horizontal gene transfer. Horizontal gene transfer is the opposite of vertical evolution. In vertical evolution, we have passing of genes from parent to offspring. In horizontal gene transfer, we have gene transfer from one organism to the other without that reproduction aspect. So we have gene transfer between organisms or between species that are unrelated to one another. So horizontal gene transfer is any process where an organism gains genetic material from another one without being the offspring. So parental inheritance is vertical evolution, horizontal is across species. I'll show you an example in just a minute. So our tree of life, our big phylogenetic tree of life, is really a web of life. All of the trees that we've looked at are showing vertical transfer from that common ancestor due to homology, due to descent with modification. But horizontal gene transfer has actually played a significant role in many of the different taxon that we have in our tree of life. And if we go back to thinking about early life as being protists, prokaryotes being bacteria, being archaea. Prokaryotes have can and still do share genetic information across species. Likewise, our early eukaryotic cells were single-celled organisms that probably experienced horizontal gene transfer as well. So currently, horizontal gene transfer is more prevalent among prokaryotes. We will learn about the mechanisms when we get to chapter 17 and we talk about bacterial gene transfer. Horizontal gene transfer is less common in eukaryotes, and that's because eukaryotes tend to reproduce sexually, meaning you need a second genome. You need a sperm to fertilize an egg or the equivalent. So many of our eukaryotes are not doing horizontal gene transfer anymore because they are more complex organisms. But when we get to chapter 17, we'll talk about the 3 ways that horizontal gene transfer occurs: conjugation, transformation, and transduction. You don't need to worry about them now. An example of horizontal gene transfer is sharing of antibiotic resistance among bacteria in a healthcare setting. So this is something that many of you are probably familiar with. You've probably at least heard of sharing of bacterial antibiotic resistance. And this is due to one of those mechanisms of horizontal gene transfer. So 2 bacterium that may not be the same species, or maybe different populations of the same species, can share genes with one another. So horizontal gene transfer was likely prevalent in that universal common ancestor. And when we think about horizontal gene transfer being important, we can imagine that there may actually have been a whole community of single-celled organisms. And so it may not have been the case that a single ancestor, a single cell, is the ancestor to all. What could have potentially happened is that there were a variety of ancestral cells, and they began to form and then evolve, and then began to transfer genetic information across those groups. 2 examples of horizontal gene transfer that you are likely familiar with are the transfer of genes in the evolution of the mitochondria. So if you remember back to Biology 101, we talked about the endosymbiont theory, where a larger host cell engulfed a smaller bacterial cell, and that bacterial cell evolved into the mitochondria. Mitochondria still have their own DNA. So that is an example of bacterial gene transfer from a bacteria to an archaeal ancestor, some species from the archaeal lineage, and that DNA becoming incorporated into our eukaryotic group. In a second transfer, the precursors to our plant cells also acquired the precursor to a chloroplast. The chloroplast was originally derived from a bacterial cell, and that bacterial cell transferred its DNA to the plant lineage. You can see many other examples here. They're not labeled specifically, but there are other potential places where we could have seen horizontal gene transfer.